still in cell structure. Yes, we're going to be here for a little bit longer. Hey, welcome back. This is actually section 2. So, section 2 is cell structure. If you remember previously, we've talked about microscopes, we've talked about the cell components. Now we're on to one specific cell component. We're going to talk about the barrier. The barrier is really important. That's aka cell membrane. There's so many important components to that, so I really want you guys to hopefully get a good grasp of how stinking important it is by all the different things we're going to talk about, okay? So let's go into it just a little bit further. So the barrier, if you notice, there's a lot to talk about. Cell membranes rock. You know why? Cell membranes are the protective barrier between the internal part of the cell and the external part of the cell. That's so important and so vital because obviously what happens internally can be greatly affected by the external components. So make sure that you guys understand the importance of it. That's why we're spending an entire section on it, okay? So, cell membranes rock. Yes, it is the gateway to the cell. Alright, so let's go over a couple things. First of all, I want you guys to notice this is actually what the cell membrane looks like. It's not just this kind of crazy single layer of whatever. It's a very specific layer. We call it, or a very specific membrane, very specific components involved in it. We call it a lipid bilayer. Lipid because there are lipids here. These yellow things are lipids, okay? And underneath attached to it are little phosphates. So we call it a phospholipid bilayer. Bilayer means two, right? There's two. So the cell membrane is actually made up of two layers of these lipids, okay? And then you see these lovely purple things. These are proteins that are embedded within the cell membrane. And as we talk about in just a few moments, these proteins are very important as they allow things to come in and out of the cell, okay? So if you notice, this is the inside of the cell membrane where the cytoplasm is and where you'll find all of the organelles. And this is the outside of the cell, okay? So, again, huge barrier. It protects things inside the cell. It allows things to get out that need to get out, and so forth, okay? So remember this structure as we talk about a couple of the things. So, the basics, okay? The basics are the cell membrane separates living cell from non-living surroundings. Seems pretty basic, right? Okay, well, let's go into that a little bit further. Again, another picture of the phospholipid bilayer with the lovely proteins, and we'll talk about that function, but just keep giving you some visuals so you guys know what we're talking about. Okay, it controls traffic in and out of the cell. So things need to get out, things need to get in, okay? And it's very important when we have something that can control that. We just don't want things kind of having their minds of their own and, hey, I think I'm going to go into this cell and I think I'm going to leave. So no, it's not that way. It's very regulated, okay? Also made of phospholipids and proteins and other macromolecules. We're not going to go into the details of all of these things, as I want you to know the phospholipids, those play an important part, and the proteins play a huge part, but there are also many other things that are actually embedded within this cell membrane, okay? That is a thin barrier, okay? It's only 8 nanometers thick. I need to give you an example of what that is, but very, very thin. Think of a microscopic cell. We can't see it without a microscope, so you got to believe that that cell membrane is ultra thin, ultra, but even though it's thin, it plays a pretty protective role, and it's pretty amazing in what it does. Okay, so it is selectively permeable, and that's in orange because it's important. It allows some substances to cross more easily than others. So, say like you're selective about, who am I going to date? Do I want to date blondes? Do I want to date redheads? Do I want to date? That's what we mean by selective. Are you going to be picky with what you bring into your house and meet your parents? So, same with the cell membrane. It's very selective about what can cross through that membrane, okay? So it doesn't just allow anything, which is a good thing because that could be pretty detrimental. Let's break it down even further. So remember, I've talked about the phospholipids and the fatty acids, okay? So basically we have the phosphate group, which we consider that head, and that is a hydrophilic, okay? hydro means water. If you break that apart, philic means loving. So it loves water. Woohoo, loves water. But these fatty acids, which we call the tails, okay, let me see if we kind of move in here. So the fatty acids are hydrophobic. Hydro, water, phobic, fearing. So when you guys think about if you add water and oil together, what happens? They separate. That's kind of an example of what hydrophobic is. It's going to, I don't want to be near you. I'm not going to mix with you. So the fatty acids, the tails, are hydrophobic. Okay? So again, here's that phosphate molecule, and then you have the tail, which are those lipids, okay, those fatty acids. So this part, phosphates, love water, okay, hydrophilic, while the tails, the fatty acids, they don't want to be near water, okay, they're going to repel water, okay. All right, now, arranged as a bilayer. So there's two layers that basically, they're not one on top of the other, but one is kind of flipped upside down, and they kind of sandwich each other, okay. And so basically, you have all those fatty acid tails. Let's go back to this picture here. You have all of these fatty acid tails that are together. So this area right here is very hydrophobic. No water. Don't want water in here. Okay, water molecules, stay away. Repel from that. But out here, again, these phosphates are hydrophilic. So they're like, yeah, water, come on closer. Okay, so inside of the cell and then outside of the cell. So you have that phospholipid bilayer. Okay. Now let's talk a little bit about how things get in and out of the cell. There are three main ways. We're going to start off with diffusion, okay? Diffusion is something that you guys probably have heard of before. We also kind of refer to diffusion sometimes, well, I shouldn't say sometimes, always when we're talking about diffusion with water, we call that osmosis. So the big thing is diffusion is the same as osmosis, but basically you're moving from a high to a low concentration. Okay, and we also call this passive transport. The reason why we call it passive transport is because basically it'll do it without anything saying go ahead and do it, or with any energy putting towards it. Okay, so passive means it's going to happen all the time. Diffusion is happening constantly. Anytime you have a higher concentration somewhere and then say somewhere else there's a lower concentration, you're going to have diffusion. Okay? No energy is needed for this, which is a great thing, so it's constantly happening. So I want to give you an example before we look at this picture here. Okay, so I always think about this. I used to be a huge concert goer. I'd go to concerts anytime I could. I'd save up my money from my jobs, and I'd go to concert after concert. And which is funny, I don't really like crowds now, but I liked crowds back then. So I was always going against the diffusion method, which is typically if you have this concert, you have these people jam-packed, and most people just want to break free out into where there's less people. That would be a good example of diffusion. I, on the other hand, I was a rebel. I wanted to stay in that pack, okay? So that's not normal. That's not diffusion. Typically when you're in a tight spot, you know, especially if it's something you're not enjoying, but you know, if you're in a tight spot and you're surrounded by people, you just want to get to a place where there's less people. That's diffusion. Higher concentration, get me out of here. Lower concentration. But then here's me. Yeah, woohoo! Okay, yeah, that was me back in the day. Now, no thanks. I don't like crowds. Okay, so here's a good picture of diffusion. So take, for instance, you take some food coloring and you drop it into some water. Okay, so you have these red molecules here showing down here. And all of a sudden, and by the way, these molecules happen to be hydrophilic, so they're going to blend in and mix well with water. Okay, so as you notice, it's going from high concentration of red molecules and it's starting to disperse itself. It's diffusing out into the water molecules until you have this kind of tinge of red in which the red molecules, the dye molecules, have basically diffused with all of the, the area that they can. It's evenly distributed, as it says up here. Okay, so diffusion basically starts off as this high concentration. I want out, so now you're moving over to this area where you're all dispersed. Okay, so that's an easy way to view diffusion. That happens all the time, remember? Okay, now, how can I get through to you? Well, so another way that we get things through the cell membrane, okay, now something called passive, oh, I'm sorry, I don't know what I'm thinking about. And I gotta plug in my computer. So basically passive transport, simple diffusion, kind of exactly what we talked about. Okay, so passive transport, let's give some more examples of that. Simple diffusion, basically diffusion of nonpolar and hydrophobic molecules, goes from high concentration to a lower concentration gradient. I guess I just wanted to reiterate everything. Sorry, I guess I forgot I put this slide in, but good, it gives us yet another chance to discuss it, right? Always ways to keep talking about stuff. So simple diffusion, so we have lots of molecules out here, okay, high concentration, so obviously we're going to have to diffuse to the place where there's lower concentration. So it's going to move through the cell membrane, and it's basically going to go to the place where there's less concentration. So materials move down their concentration gradient through the phospholipid bilayer. Okay. Now again, back here, if you look, diffusion of nonpolar hydrophobic molecules only. Okay. So hydrophilic is going to kind of chill out in here. We don't want that. Diffusion has to be nonpolar and hydrophobic molecules. So that's why this can happen kind of at any time, only with these types of molecules. Okay. So there's simple diffusion. Passive transport. Okay. Passive doesn't need energy. All right. Now here's another way. How about facilitated transport? Okay. Here we're going to talk a little bit more about those molecules, lovely proteins that you saw embedded in the phospholipid bilayer. Okay. Diffusion of polar hydrophilic molecules. Okay. So it is diffusion still. So we're going from high concentration to lower concentration. But now we're talking about polar molecules and hydrophilic molecules. Molecules that love the water. Okay. But here we need to actually go through a protein channel. And that's very important. It's not just diffusing through the membrane. It actually needs a channel to go through, because otherwise, remember, if we're dealing with hydrophilic, it's going to just kind of chill out, you know, in there. We're going to love those water molecules. We're not going to have that easy or the easy diffusion through the membrane. So instead, we got to use this protein channel. So here's a picture of it, where basically you have facilitated diffusion, okay, and you actually use this protein molecule. So the passage of materials is aided by both a concentration gradient and by a transport protein. Okay, so you have the aid of this protein to get some of these molecules through that cell membrane. Okay, so remember, hydrophilic is going to have a hard time with these fatty acids. Okay, because remember, those fatty acids are hydrophobic, so it would have a hard time passing through. So for that reason, we have to use some kind of a transport molecule. Okay, in that case, it's a protein. All right, now last chance to get through the cell membrane. Here is one of the last ways that things can move in and out of the cell. It's called active transport. Okay, diffusion against concentration gradient. So diffusion. Remember, I was telling you about the concerts and how I was the rebel. Well, guess what? That's what active transport is. Active transport is diffusion against the concentration gradient. So we're going from, oh, I have all this room in the world, to I need to be at the stage, rushing it, being up there with all those people. Okay? So that's what active transport is like. You are going against the concentration gradient. But in order to do that, you have to use a protein pump. Okay? So kind of like facilitated diffusion, in that you used a protein in order to get things through. But more importantly, you have to use ATP. So if you notice this, okay, active transport is going against the concentration gradient. It's going against the grain. Okay, you're being a rebel. And in order to be a rebel, it takes energy, right? If any of you guys are rebels, you know what I'm talking about. So molecules, again, move through a transport protein, but now energy must be expended to move them against the concentration gradient. So we need ATP in order to do that. Okay? So again, we know that ATP is produced in the cell. Where? Yes, the mitochondria. Good job. I bet someone hopefully said that. So ATP is produced in the cell, and some of it is actually used in the cell in order to get things against that concentration gradient. Okay? Whether it's inside or outside of the cell, moving stuff inside or outside. Okay? So a big thing, actually I want to go back here and just pull back a little bit. Let's make this smaller. An orange requires ATP. Okay? So the big thing is diffusion, doesn't take any energy. Facilitated diffusion doesn't take any energy. But active transport does, because you're going against the grain. Okay? So let's do a little recap. How about a little review? So let's look at this picture again. This is showing all three different types. You have simple diffusion, basically here, higher concentration to lower. Then you have facilitated diffusion, where you're still going from higher concentration to lower, but we're dealing with hydrophilic, okay? Hydrophilic molecules and polar molecules that basically resist this cell membrane, okay? So you have to use a protein for it. Passive transport, both of these are passive transport, so you're not using any energy. Active transport, we're going from lower concentration to higher concentration. And because of that, you're using a lot of energy. Maybe not a lot, but you're using energy, okay? So that's the big difference between those three things. One's going to take energy, the other two are not, but two of them require protein channels, okay? So there's a little bit about the cell membrane. Watch this for some visuals.
The smallest unit of life is the cell. In order for life to be sustained, nutrients and gases must be able to pass into a cell. Wastes and other products must be able to pass out of the cell. Many of the materials that pass in or out of the cell do so by passive transport. In this process, there is no energy, no ATP expenditure by the cell. Concentration gradients allow for the movement of materials. Osmosis and dialysis, both specific types of passive transport, use this concentration gradient of high to low. Another type of passive transport is facilitated diffusion. Since it is passive, the high to low concentration gradient is present. However, the process is facilitated by a carrier protein. The carrier protein is somewhat like a revolving door, allowing easy access across the membrane and then rotating back to the other side. All of these processes, osmosis, dialysis, and facilitated diffusion, are passive. The cell does not have to use any energy to move materials by these methods. The concentration gradient is used. However, there are materials that need to be moved against their concentration gradient. This process requires energy, ATP, and protein carriers that are found in the plasma membrane. Defined active transport is material moved from a low concentration to a high concentration through a membrane using a protein carrier that requires ATP. An example of active transport is the mechanism of uptake of iodine by the thyroid gland. Iodine is essential for the manufacture of hormones that influence body metabolism. When you eat a meal containing iodine, the iodine will pass from the small intestine into the circulatory system. As the capillaries pass through the thyroid gland, iodine diffuses out of the circulatory system. Iodine attaches to protein carriers in the thyroid cell's plasma membrane. Being proteins, they have a unique shape, so only iodine will fit. Glucose, which is also actively transported into the cell, relies on a different protein receptor. The energy required is somewhat analogous to a rock being pushed uphill. The processes of simple diffusion, osmosis and dialysis, facilitated diffusion, and active transport allow for most all of the small material, those dissolved in plasma and extracellular fluid, to pass in or out of the cell. Large particles, however, pass into the cell by endocytosis. This is an active process. It needs ATP. In this process, the plasma membrane surrounds the particle, such as bacteria or a food morsel, and forms a vesicle that pinches off from the membrane. The cell does this in two different ways. One way is called phagocytosis. In this method of cell eating, large particles are surrounded when the cell extends the plasma membrane and engulfs the particle. The result is a membrane-bound package of material. Fluids that the cell wants to take in enter by a process called penocytosis. In this type of endocytosis, the cell membrane sinks in, the material sinks inward, and the membrane pitches off. The cell uses energy for this movement. In both types of endocytosis, the cell ends up with a membrane-bound vesicle. The material in the vesicle still has to pass through the membrane in order to be used by the cell. In this case, the lysosome joins with the vesicle and digestive enzymes break down the material to particles small enough to pass through the membrane and into the cytosol. A specialized endocytosis called receptor-mediated endocytosis also occurs in cells. In this process, molecules of a particular substance, such as cholesterol, bind to receptors in the plasma membrane. The vesicle is formed as the membrane sinks in. Then a lysosome joins the vesicle and the digestive enzymes break down the substance, releasing it from the receptors. The substance diffuses into the cytoplasm, the lysosome reforms, and the vesicle, with now empty receptors, becomes part of the plasma membrane again. This allows for the cell to reuse or recycle the receptors. A function of some cells is to manufacture or synthesize materials for export. In many cases, this material is made up of large molecules like proteins that cannot pass through a plasma membrane. The process of exocytosis enables the cell to pass this material out of the cell. The cell is a very dynamic living entity. It is essential that materials move in and out of the cell. This is done by passive transport using a concentration gradient, and active transport, endocytosis and exocytosis, using ATP.
So there you have a great video that I haven't seen it since last year, and so I'm watching it and I'm like, wow, I forgot it touched on some really great things that are in your lesson that I didn't really go into depth. Endocytosis is a huge part of getting things in and out of the cell. And I think I even mentioned the lysosomes. Obviously, that is one of the organelles within the cell that usually breaks down old, kind of worn out material within the cell, but it plays a huge part in basically allowing things into the cell via phagocytosis and pinocytosis. It breaks down kind of that membrane and then breaks those particles into smaller things. So I'm really glad that that video touched on that, because I didn't go over that specifically because I wanted to make sure that you guys got that out of your lesson as well. I'm not going to take everything from your lesson and go through it, but make sure that you guys go through that a little bit. So that's a great review. In the cell membrane, obviously, you've learned it's very important as things come in and out, some easily and some not, but all vital. So there you have the barrier, and we are actually done with section 1 and section 2. Okay, so when we come back, we're still going to be on cell structure, but you guys will notice that we will switch gears a little bit and go into cell reproduction. Okay, so now my daughter's playing with my phone. Lovely, right? Okay, well, you guys all have a wonderful day or night, depending on when you're watching this, and we'll see you back here for section 3, and that's it. It's a wrap! Woohoo!